everyone to the Greener Hub webinar series. My name is Evelyn Zuniga, and I'm one of the communications officers for the Greener Hub project. The Greener Hub project officially started in September 2022 as a coordination support action of the European Commission under the Horizon Europe program. It currently represents 15 ERANET co-funds and self-sustained initiatives in the field of agri-food and biotechnology. We started these breakfast clubs last year in October, and the topic of our first series was circularity in agriculture. So if you're interested in topics related to mixed farming systems, mitigation of greenhouse gas emissions, sustainable farming, circular food systems, all our recordings are available now in YouTube. Now and for these 3 months, we are going to focus on a different topic, which is going to be biotechnology to advance sustainable solutions for bioeconomy. And we are going to have topics related to plastic degradation, production of chemicals in a sustainable way, biofuel production, protein engineering, and among many others. Just if you have housekeeping announcement, please feel free to use the Q&A box and the chat box to put your questions. I'll do my best to address as many as I can with the time that we have, which is 1 hour. Okay, so let's get started. Today we have 2 panelists, Professor Frank Delvigne and Dr. Alan Goddard. We are going to open our round of presentations with Professor Delvigne, who is a professor of microbial biotechnology from the University of Liège. His research focuses on topics like identification of the factors affecting microbial population physiology in the context of bioreactor scale up or down, the development of innovative single-cell technologies for controlling microbial population heterogeneity through cybergenetics approaches, online flow cytometry and time-lapse imaging with feedback control, as well as study related to a better understanding of functionality of biological noise in the context of microbial populations used in various bioprocess applications. Today, Professor Delvigne's talk is entitled Control of Yeast-Bacteria Co-Cultures under Continuous Cultivation, which is part of the Bio project from the ERACO Biotech initiative. Professor Delvigne, thank you very much for joining us today and for accepting our invitation to present your project. And now you can start your presentation. The floor is yours. Thank you so much.
Pointer. Okay. As said into the introduction, the main topic we have in my research team is to address this microbial population dynamics, cell population dynamics, and how to make new bioprocesses out of it. I will start first with a small introduction about my working environment research topic that will help you to understand this, let's say, application about yeast-bacteria co-culture. In fact, since a couple of years, together with my colleagues, we have developed a concept to capture the main features behind cell population dynamics. And we call this concept cellular entropy. You have a lot of synonyms into the literature. You have phenotypic heterogeneity, biological noise. This entropy refers to cell-to-cell differences in main phenotypic features. Okay, this low entropy means that you have a quite homogeneous population, which is expected for most of the bioengineering application. And the high entropy, on the other hand, means that you have a huge difference, cell-to-cell difference in phenotypic features like growth, like gene expression, like metabolic activities, and so on and so forth. Okay, this entropy is a keyword for this presentation, and I will show you its importance in the context of stabilization of microbial cell population, and for application in the context of industrial biotechnology. Actually, we have 4 main lines of research. The first one, and I will do, I have 7 slides about this. It's half of my presentation, is the control of temporal gene expression profile, which is at the basis of our main discoveries about cell population dynamics. That's why I will spend a bit of time on this. Then we have the control of complex phenotypes. By complex phenotypes, I mean sporulation, biofilm formation, complex stress response pathways, and so on and so forth. We have a more recent focus also on protein and product secretion from a cell to another. And the topic of today, that will be the second half of my presentation, is about co-culture and communities control. Okay. Besides these applied topics, we have also a lot of methodological development, and we spend a lot of time in automating flow cytometry in order to be able to, analyze cell population features in almost real time. Okay, you probably know that in this field, the rock star is microfluidics, in fact, and indeed in microfluidics you can extract a lot of single cell features. We are also using microfluidics in our team, but we do prefer using automated flow cytometry because of the experimental throughput of these techniques. And also, as I will show you, this automated flow cytometry can also be directly adapted on a standard cultivation device that will also ensure the scalability of your results. Thus we have, applied research topic, we have methodology development, and also we spent a significant part of our time on fundamental research. And the goal again is to, what we try to do out of all this application is also to find common features on, let's say, main factors driving cell population dynamics. I have listed here the most important factors, and we recently discovered one which is really important, which we call the switching cost. I will give you details about this in a minute. And these main drivers, in fact, sustained a principle which is called growth entropy compensation, which we think is at the basis of most of the cell population development features. Okay, we'll see that we'll be focused mainly on microbial population, gram-negative, gram-positive bacteria, as well as yeast. But we do foresee also that this type of principle also could stand for higher eukaryotic cells. Okay, but that's for the future. Okay, I will start with a basic description of dynamics of cell population. Okay, let's say that you have a cell population evolving in an ecosystem in this case. And if you speak with people working in the field of microbial ecology, they will tell you that, okay, having a heterogeneous population that's displaying high entropy is something which is nice because that will enhance the resilience of the microbial population to environmental fluctuation arising into this ecosystem. Okay. On the other hand, when you speak with bioengineer, it's the totally opposite. In fact, when you are engineering gene circuits, you put the gene circuit in cell population to produce something interesting out of it. What an engineer is expecting is to have a quite homogeneous response at the level of the gene circuits, this low entropy, in order to enhance predictability about the fate of the bioprocess, control over the bioprocess, and stability of the microbial population. Okay. What I will show you today as an introduction is that, okay, the truth is in between these two views, in fact. And the main take-home message is that, okay, we should pay more attention at the level of the cell entropy in order to be able to control more efficiently cell population for given bioprocesses. And I will show you an example. As I told you in the introduction, most of our experiments are done with automated flow cytometry. We have designed softwares, we have designed also physical interface, allowing us to connect directly the flow cytometer on cultivation device. It can be a shake flask, that can be these fancy key bioreactors or standard bioreactor systems. And each 5 to 10 minutes, you can take a samples, dilute it, and trigger the flow cytometry analysis. Okay. Since we are also analyzing the data online, I will show you also that further on we'll be also able to do feedback control based on the flow cytometry data. Okay. And out of this, okay, you can have these very nice movies. Because here you have an example on the evolution of an E. coli population, Saccharomyces cerevisiae population, and Bacillus subtilis population. with different phenotypic features. And this is how the systems look like in reality. There's a lot of tubes, a bit messy, but it's very effective to really get data in a fast and affordable and reliable way. Okay? Of course, instead of these movies, we can process the data in order to have quantitative stuff. I will not go into the details because that's not the point of this presentation. But to make a long story short, since 5-6 years now, we had the opportunity to investigate a lot of different cellular systems. Like I said, different type of bacteria, E. coli, Pseudomonas, Bacillus, a bit more exotic bacteria also, as well as yeast, Saccharomyces and Kazakhstania yeast. And when we compare all the data gathered by automated flow cytometry, we have been able, in fact, to classify the different diversification profile of this cellular system. Okay, because this is a typical display you have out of an automated flow cytometry experiment. This is the time. Sorry, I didn't put the label, but this is the time, and this is the fluorescence intensity. And okay, if it is dark, you have a very few cells. And if it is bright, in fact, you have most of the cells that are displaying this fluorescence intensity, okay? And what I didn't tell you also is that in order to be able to investigate phenotypic features based on automated flow cytometry, we are using fluorescent reporter, okay? We have very different type of gene circuits that have been already investigated, natural and synthetic, and this classification comes from this. Okay, I would propose not to go into the details at this stage. I will show you an example, more concrete example in the next slide. But what do we have when we compare all the cellular systems is that, in fact, we can see that we have an evolution of the diversification profile according to what we call the switching cost. Okay, what is the switching cost? But let's say that you have a cell with a gene circuits, and you know that cells can activate gene circuits to adapt to new environmental conditions. Or if we are in the context of bioprocess, you can also engineer your cell gene circuits to make cells produce something interesting out of it. Okay, and when cells decide or is forced to activate the gene circuits, it can display an alternative cell diversification pattern. Okay, and the first one we have is what we call the constraint diversification pattern. In this pattern, you can see that most of the cells are following the same path. Okay, few cells didn't activate the gene circuits, and if we put, in fact, the diversity expressed as entropy in function of the growth rate, you can see that when we switch from the off state to the on state, in fact, you have almost no difference at the level of the growth rate of the microbes, and you have no difference at the level of the entropy of the population. Okay, the entropy here, the dispersion of the population here and here is approximately the same. What we have observed, on the other hand, is that when the activation of the gene circuits leads to a loss in growth rate at the level of the cell population, you can see that this loss of growth rate is compensated by a gain in entropy, a gain in the diversity at the level of the activation level of the target gene circuits. This is what we call growth entropy compensation. And we have this very nice diversification profile. As you can see here, that we have a far more diversified population by comparison to what we have here. We call it this dispersed diversification. And for very extreme switching costs, we have also this bursty diversification pattern. That you have almost all the cells that are not activating the target gene circuits. But from time to time, you can see that you have wave of diversification. And the cells that get activated, in fact, lost totally the capability to grow and are washed out if you are working in continuous cultivation devices. Okay, and this is very nice because then, based on this very simple parameter, you can predict which type of diversification profile you can expect out of the cell population. Also, based on this diversification profile, you can predict what will be the level of controllability of your systems, the level to which you will be able to manipulate the level of activation of your gene circuits externally based on environmental stimuli, such as additional nutrient, additional inducer, or change in physicochemical conditions such as temperature and pH if you are working in bioreactor. Okay? And for this, in fact, we have designed a system. We call it the segregostat. It has to be connected to a continuous cultivation device. We can take benefit of the automated flow cytometry to monitor online the state of the cell population, but also you can act on the cell population by adding or changing the environmental condition. Okay, I will show you an example and then I will switch to the co-culture topic. Here we have investigated a system in E. coli, which is a system which is widely used to produce recombinant protein in industry, which is the T7-based expression vector. And based on this vector, in fact, you can rely on the T7 RNA polymerase that will take the control of the host to divert the resource out of this host and lead all this resource towards the production of the target recombinant protein. Okay, and you can imagine that when you activate such circuits, there is a huge cost, a huge switching cost over the cellular system. Okay, and these are the data we have. These are preliminary data. The manuscript is in preparation. Here we have used the segregostat to be able to control the level of induction. Okay, you have a first induction of the system. Okay, you can see that almost all the cells get induced, but then since this induction leads to a loss in growth rate, you can see that we generate the cell population automatically generate diversity, generate entropy to compensate and to adjust the cellular density into the bioreactor. Okay, and then you can do that at a given frequency. And of course, if you are working in bioprocess, what you want to do is to keep almost all the cells into these highly activated states. And in order to do so, you can also use the segregostat and you can push the cell population based on more constrained, control rules, leading to an increase at the level of the frequency of activation of the cell population. Okay, and what you can see, okay, it works. But then for a given frequency, in fact, the entropy compensation stuff is not able to be installed to stabilize the cell population. And you can see here that the cell population escape this forcing by exhibiting an intermediate phenotypic states with a lower activation level and a lower, let's say, impact on the cellular growth. Okay. What can we learn out of this for setting up new bioprocess? In fact, this is the classical view about how to design a bioprocess. That you have typically a growth phase where you increase the cell density. Then you activate target gene circuits to induce the production. And during the production, in fact, the population totally collapses because of the switching cost. Okay. Actually, we are able to propose a new strategies where we are able, based on automated device, to alternate between growth and production and to stabilize the population, further enabling the establishment of continuous bioprocess in the field of industrial biotechnology. And based on this, okay, the next question we ask is that, can we adapt the same, let's say, principle in order to control two genotypes instead of phenotypes? Okay, so can we apply the same device to control the composition of co-culture in real time? Okay, by alternating between conditions favorable for species, a first species, and conditions favorable for second species, for example. And okay, we have been funded by the Biotech to do so. And during this Biotech project, we have investigated different types of yeast-bacteria co-culture. Why yeast-bacteria is because then you're able to combine the biochemical potential of eukaryote cells and prokaryote ones. But also, if you are using flow cytometry, it's very easy to make the distinction between yeast and bacteria because of the cell size. Okay, this is a typical flow cytometry analysis with the 4 scatter and size scatter, very basic parameters in flow cytometry. And you can clearly distinguish between the yeast population and the bacterial population. Okay, and then you can directly apply our segregosta concept without the need of fancy fluorescent reporter, leaving the room for further development in the field of synthetic biology at this level. What we have been considering to be able to control co-culture is principle was well established in the field of microbial ecology, which is the concept of competitive exclusion and the concept of metabolic niche. Okay, that's stating that if you have only one metabolic niche, and okay, in the context of microbiology, it is translated into if you have only one carbon source available for the growth of microbes, you have only one species that can dominate. Okay, if you want to stabilize a co-culture, then you need at least 2 carbon sources. Okay, and then you have tried to apply this concept to our segregosta device. And what we have decided is to be able to identify metabolic niches for the bacteria and metabolic niches for the yeast, and to use the segregosta to alternate at the specific frequency between these 2 metabolic niches, further allowing the stabilization of the composition of the co-culture. Okay? This is the ideal case where the 2 metabolic niches are totally separated. But in the real case, and I will show you some example afterwards, these metabolic niches can from time to time also overlap with each other. For example, the yeast can produce overflow metabolites that can lead to dioxide shift, promoting the growth of the yeast and the bacteria at the same time, okay? Making the control a bit more difficult to apply. Okay, let's go to the case studies. We have first considered a very simple co-culture, with the yeast being Kazakhstania bullderi and the bacteria, sorry, being Lactobacillus plantarum. It's very easy because we know that yeast and lactic acid bacteria have co-evolved in a lot of different ecosystems. And actually, if you look at the genome of most of the lactic acid bacteria, you can see that there are some important genes lacking for the self-sustaining growth of the lactic acid bacteria, such as genes involved in essential amino acid synthesis of ribonucleic acid synthesis. And in fact, this lactic acid bacteria can take benefit of amino acids released by the yeast itself in order to be able to grow. Okay, and then we have this cross-feeding, which is also known in the field of microbial ecology to be very useful to stabilize natural co-culture. Okay, what is even more interesting to stabilize co-culture is mutual cross-feeding. This meaning that the yeast produce something useful for the bacteria, but also the bacteria produce something useful for the yeast. Okay, and then we try to install this mutual cross-feeding by pulsing maltose into the system, the pulse of maltose being automatically triggered by your segregosta platform. And what is nice with maltose is that maltose is not consumed by Kazakhstania, but it's processed by Lactobacillus plantarum, and it's breakdown into 2 glucose molecules. And one of these glucose molecules is phosphorylated and used by the bacteria, but the other molecule is released and can be used by the yeast. Okay, thus by pulsing maltose, we have been able to close the loop and to provide this mutual cross-feeding to this synthetic community. These are the results. Okay, thus without control first here, thus we have the population of Kazakhstania in blue and the population of Lactobacillus plantarum in orange. And you can see that even without control, at the end, the culture tend to self-stabilize. Okay. Of course, when we apply control, we call it automated reversion of metabolic niches. you can see that we can stabilize very efficiently the co-culture. Okay. Then I will move at the same time because I'm out of battery, but I will continue my talk, don't worry. Then let's try to consider something which is a bit more complicated, and that will lead to the end of my talk. Okay, let's try to consider two microbes that are of interest for industrial biotechnology, Saccharomyces cerevisiae and E. coli. Okay, but here we don't have this mutual cross-feeding, but we expect to have a bit more, let's say, difficulties in order to be able to stabilize this very interesting, potentially interesting co-culture. Okay, what we know is that both the bacteria and the yeast in this case can consume glucose. Okay, this applies glucose pulsing, but also both of these yeast and bacteria are able also to exhibit overflow metabolism. Okay, thus when we have a lot of glucose, part of this glucose is diverted into acetate for E. coli and is diverted to ethanol in the case of Saccharomyces cerevisiae. And what we have observed during preliminary experiment is that, of course, if we put only glucose, the bacteria wins. Okay. But if we give enough time to the system to get some relief, in fact, we can see that when we have a diaxic shift on acetate and ethanol, the yeast is more competitive at this level. Okay. And then here we use the segregosa to alternate between glucose pulsing and ethanol pulsing in order to be able to adjust the co-culture composition. And these are the data. And as without control, you can see that it's very difficult, in fact, to stabilize the co-culture. You have E. coli in orange here that wins all the time, almost all the time. And when we apply our control procedure, this automated reversion of metabolic niches, it works. You can see that the systems try from time to time to escape the control, but okay, it works. Okay. And then based on this, we decided to go for a process of interest, a process of coumaric acid production. And in this process, in fact, we decided to establish a division of labor mechanism between E. coli and Saccharomyces cerevisiae. The E. coli is doing the first step. The use carbon source to produce tyrosine. And tyrosine is bioconverted into p-coumaric acid, the molecule of interest by Saccharomyces cerevisiae. Okay. We have not been able to find this mutual cross-feeding for E. coli and Saccharomyces cerevisiae. And instead, we use another concept in order to ease the stabilization of the co-culture, which is also widely used in synthetic biology, which is called the separation of the carbon sources. And here what we did is that we modified the E. coli to make E. coli unable to use glucose, okay? But of course, E. coli is still able to use xylose, and you know that xylose is not processed by Saccharomyces cerevisiae. Okay, thus we try to specialize each of the species into the utilization of a specific carbon source. Here are the data. This is without control. That's very similar stuff that we had before. And with feedback control, you can see that okay, we can smooth a bit more the stabilization of the co-culture. But even if we use the separation of the carbon source, you can see that okay, we are not really able to maintain perfectly the 50-50 stabilization during the whole time. It's not due to the competition for the carbon source, but it is due to the accumulation of coumaric acid into the extracellular medium. Coumaric acid exerting some inhibition on Saccharomyces cerevisiae and E. coli in our case. Okay. Then in the future, we plan to improve our control procedures in order to refine the stabilization of this type of co-culture. Even though we have been able to reach decent titer of p-coumaric acid with this approach, we are at 1 gram per liter per hour, which is not bad by comparison to what is actually proposed in the literature. But more importantly, again, this is also a process which is done in continuous mode. that is more easy to be upscaled at industrial level, and is also more productive than classical batch and fed batch bioprocesses. Okay. By this, I arrive at the end of my presentation. Of course, all this work have been done by a very nice team of motivated people. You have the name listed here. I have also a nice collaborator in Bielefeld, Karlsruhe, Leipzig, and Mexico. And I'm also grateful to the ERACO Biotech that allowed us to get this very nice data about culture dynamics and culture control. Thank you very much.
Thank you very much, Professor Delvigne. Very interesting presentation. I invite everyone in the audience to put your questions in the Q&A section or in the chat section. In the meantime, I have a question. So when you were talking the first part of your talk about the GFP reporter and then kind of checking how it stabilizes, did you use, it was like hooked to a constitutive promoter, or it was an inducible promoter to check?
In this case, yeah, most of the time it's inducible promoter, but it depends on the context. Now we have analyzed sporulation networks, general stress response, activation of alternative carbon source gene circuits, and so on and so forth. This every time it's more inducible than constitutive. And in this case, okay, it's a purely inducible system, and the T7 systems is based on the lactose-inducible promoter. And then when you add lactose, the RNA polymerase of the T7 phage is synthesized and take the control over the host for the production of the target recombinant protein. But that's only one example. We have designed a lot of different gene circuit, inducible gene circuits with different reporter systems. And all these systems are described in this publication if you want to have the details.
And we have a question here in the Q&A section. It says, very nice talk. Thank you. How does ARMN or feedback control finally work?
I will go back here. You have flow setup, that's okay. Let's say that each 5 minutes, you are able to analyze your cell population based on flow cytometry. That's based on this, you can easily compute the ratio between the yeast and the bacteria. And then let's assume that you want to maintain 50% of yeast and 50% of bacteria. That is your control set point. And you have identified the metabolic niches of your yeast and the metabolic niches of your bacteria. And then based on this, it's very simple. Based on automated flow cytometry, if you take several samples, you can determine the fitness of the yeast and of the bacteria. And for example, if the fitness of the yeast is dropping down, then you switch to the metabolic niches of the yeast. Okay. On the opposite, if the fitness of bacteria is dropping down, then you switch to the metabolic niches of the bacteria. This is how the systems globally works. Actually, it's a very simple bang controller. We switch abruptly between both metabolic niches, but as I show you, most of the time you have overlap between the metabolic niches, and then in the future, we plan also to apply model predictive control in order to smooth a bit this control procedure. More specifically, this is not very useful when you have this, because it's a very easy co-culture to be done, and when you apply the control, you can see that you have this very smooth control effect. But it's more, that will be more useful in this case, where the microbial interactions and the definition of metabolic niches is a bit more difficult in this case.
How do you calculate in the co-cultivation, like the initial ratio of bacteria and yeast? I guess 50-50 or?
Yeah, it's difficult because you have a batch phase. Okay, if you switch from the batch to the continuous phase, with 50% of bacteria and yeast is more easy, of course, okay? We have been also testing different ratio, and you have ratio, depending on the case studies, you have ratio that are more prone to lead to stabilization than others, okay? That's why we decided to try to go to start at 50%. But for example, in this case, we have a tolerance of 20% over this co-culture to be able to further stabilize the yeast and the bacteria ratio in function of time. In this case, you can see that you can start with a very different ratio. It always works because you have this mutual cross-feeding effect, okay? In this case, it's a bit more sensitive, okay? There is a trick for starting the cultivation, of course.
What was my other question? Yeah, no, I think you answered. Well, I guess I have like these questions just out of curiosity, like how many, like what different disciplines you have to be, like you need to be able to work this project, because I see like there's mathematical modeling, there is engineering, there is
a lot.
Yeah, it's like
that's I have people that are more focused on molecular biology to design the gene circuits or the metabolic engineering for the different strains. I have people that are more focused on bioprocess modeling and control, and other people focused on automation also. Thus it's a mix of biology and physical systems, but it's not always easy to, let's say, to stabilize like a co-culture. But I have been lucky enough to be able to motivate people from different disciplines to work around these topics. Yes. But you have right, you really need to have different people specialized in different disciplines to be able to deliver this type of data at the end.
Yeah, it's very interesting. And so the system with tubes and then the little box that goes to the floor, all this is a closed system. You basically put everything there and then it runs until you run out of reagents.
Yeah, in fact, okay, this is here. We call it the segregostat. We just had to put a name on this by analogy to the chemostat. Thus we have the software, and this is here only the physical interface. Typically, it's simply a box with a microcontroller inside. Okay, and what we want to do also is, instead of putting patent on it, it's very difficult to patent this type of technology. In fact, you have companies that tries to do so, but unsuccessfully so far. Thus, instead, what we would like to do also is to make it open source. And actually, we are doing a lot of effort to make it available in an open science context, so that we can send the procedures to external labs, for example, or to industry. And then you are able to 3D print most of the part, or to easily get access to the electronic component of the device. And we can teach you how to assemble everything, okay? Thus, so far, we have 2 academic partners that can get access to this technology. And you have also 2 industrial partners that also adopted this technology into the R&D department.
That's fantastic. Like everyone can access it. Thank you very much, Professor Delvigne, for your presentation. And I guess we're going to move to our next presentation now. Thank you very much for everyone for your questions.
Thanks for the opportunity. Bye-bye.
Thank you. Yeah, so now let me share again my screen just real quick here. And yeah. Now we have Dr. Alan Goddard, who is a reader from the School of Biosciences at Aston University. His research focuses in the structure and function of cell membrane. He uses model lipid membranes to study proteins, receptors, transporters, and enzymes in their native environments, and also to probe the effects of various compounds in the integrity of the membrane itself. His recent research has focused on the modulation of membrane to enhance biotechnological processes. And today his talk is named Biotechnology at the Cell Membrane and is part of the MemBRane project from Eraco Biotech. Dr. Alan Goddard, thank you very much for accepting our invitation. And I'm just going to stop sharing my screen. And the floor is yours, and you can start sharing your presentation. Thanks so much.
Thank you. Hopefully you can see that presentation. Is that okay?
Yes. Yeah.
So I'm going to start by explaining the strange capital letters in the title. So the best part about doing an EU grant, in my experience, is coming up with a very forced acronym to make your project make sense. So our project is called Membrane Modulation for Bioprocess Enhancements, and all the letters in membrane somewhere come from that title. It's a big collaborative project, or was a big collaborative project. It's finished now, with a number of academic institutions. So Aston in York in the UK, Jülich in Germany, Groningen in Netherlands, and then IATA in Spain, and then a range of industrial collaborators. So MemBRane, who work on modulating cell membranes, the Nova Institute, who did all of our environmental sustainability that I'm not going to mention at all in here, so apologies to them, and then Pat Meyer and Laumann, who gave us our kind of industrial challenges that we were looking at. So I'm going to start off just simply explaining a little bit about the cell membrane, which I'm sure you all know about, but just so we're all on the same page. The cell membrane basically forms this impermeable barrier around all cells. So it's got this lipid bilayer here with the hydrophobic acyl chains in yellow and the blue head groups, which are hydrophilic. So it spontaneously forms a bilayer, which can then keep molecules out of the cell. But obviously cells need to transmit both molecules and information across that. So you've got a variety of membrane proteins that act as transporters or receptors within the membrane, and structural proteins and all kinds of things. So it's a complex mixture of different lipids and different proteins. I've worked for years on these lipids and proteins, but this project mainly focused on the lipids. And the main class of lipids that you find within cell membranes are phospholipids. Phospholipids all have a common structure. So they've all got a glycerol backbone, which is here, and then two acyl chains. So these are hydrophobic chains, which have a variety of properties I'll talk about in a minute. And they're grouped largely based on this, the identity of this head group attached to phosphate. And the head group could be something nice and simple like hydrogen, so it just becomes phosphatidic acid, or phospholipids such as phosphatidylethylamine, choline, serine, glycerol. And their main differences are the size of the head group. So, for example, if you look at choline compared to ethanolamine, it's much bigger, and the charge that's on those phospholipids. So there's a negative charge on the phosphate, which is sometimes balanced by the head group. So they're either anionic or there's vitrionic. The acyl chains themselves, as shown in these just single fatty acids, can either be saturated, which means that all these carbon bonds here are single bonds, or you can have an unsaturation in there where one or more of these carbons becomes a double bond. And what unsaturation does is it forms kinks in the acyl chains, and it reduces the interactions between these phospholipids. So what it does, it makes the membrane what we call more fluid. It means the membrane acyl chains are more easily able to move around, and molecules are more easily able to diffuse through the cell membrane. And I'll come back to that fluidity later on in the talk. So the project, and most projects in my lab, are focused on real-world industrial challenges. There were two industrial partners. I'm going to start at the bottom because that's the one we didn't actually do in this project. I'm not going to talk about today. Pat Meyer are a company who make yeast. They're based in Turkey. And as we saw in Frank's talk, you can have co-culture of yeast and lactobacillus, for example. And in our case, what we're interested in is the way that the lactic acid that lactobacillus makes can actually kill the bacteria that would normally populate a yeast fermentation. So it's used as a preservative in a yeast fermentation, but the weak acids are toxic to the cells. I'm not going to talk about that at all today because of time, but if you want to know any more about it, I'm happy to talk about it afterwards. And then Laumann are also yeast producers, and they were interested in two different stresses that yeast encounter. One of which is osmotic stress from the glucose, and again, I'm not going to talk about that, but I'm going to focus on ethanol stress at the cell membrane in yeast, and in this case, in Saccharomyces cerevisiae and related Saccharomyces strains. So how does ethanol cause toxicity? Well, pre-COVID, not many people knew about this. Now everybody knows about this because the alcohol hand wash used during COVID destroys the COVID virus because it's an enveloped virus. So it's got essentially a phospholipid membrane around it, and the ethanol gets in and disrupts and makes pores in this membrane and tears it apart. So ethanol can integrate into the cell membrane, but it can also diffuse through the cell membrane. There's not many molecules that naturally occur that actually diffuse through cell membranes because anything big and charged can't diffuse. but small polar molecules like ethanol and water, and also hydrophobic molecules like benzene and gases can diffuse through. The challenge here really is one of climate change. So climate change means you get more intense sunlight, so your grapes produce more sugar, and the yeast loves turning the sugar into alcohol. So that means your wines have a higher increased percentage of alcohol by volume. So you need yeast that can withstand that, but also yeast that can then be added to that mix later on to further ferment it to get rid of the excess sugar, for example. And also yeast that you can use to operate within the biotechnology sector for making biofuels. So we're not just trying to make stronger wine here, we're trying to make yeast that are more resistant to ethanol for a variety of different mechanisms and a variety of different processes. And the thing we targeted on was something that's actually relatively understudied because of a historical lack of tools and techniques, which is the lipid components of the cell membrane. So we took a kind of integrated approach here where you can't do everything with cell membranes just using either a kind of wet experimental approach, by which I mean the stuff we'd normally do in the lab, or even a molecular dynamics approach on its own. So we've taken the two synergistically, and we've mainly focused on the phospholipids. So we've used a few of cellular and kind of in vitro techniques to look at the effect of ethanol on cell membranes. We've also modeled them using molecular dynamic simulations, where you simulate your atoms as a bunch of particles and essentially, in my head, computational blobs that interact with each other, and you can learn things that are happening in your biological system using that molecular dynamics in silico approach. So I'm going to skip through some results, and I know I could spend hours talking about all the things we found, but I'm trying to summarize some interesting and slightly counterintuitive findings we had. So I'm going to talk about a variety of assays. I'm going to start by talking about this one, which is a liposome-based assay. So liposomes are essentially model cell membranes. They're maybe 400 nanometers in diameter, so smaller than a cell, but made of phospholipids. So they form a single bilayer, and within that bilayer, you encapsulate stuff that doesn't diffuse across a cell membrane. And as I said, most things don't diffuse across a cell membrane. And in this case, what we've done is we've got a liposome here with a blue membrane, and these stars represent the stuff we captured inside it. And the molecule we captured is this, which is 5,6-carboxyfluorocene. It's a fluorescent molecule, and the beauty of it, using it in this system, is it's actually what we call self-quenching. So it will turn off its own fluorescence at a high concentration. So if you pack it into a liposome, counterintuitively, it's not fluorescent. But if that liposome gets disrupted, so the cell membrane breaks open or there's pores created, the carboxyfluorocene will come out and you'll get an increase in fluorescence. So something like ethanol that we know damages the cell membrane should result in, if you apply it to a liposome, should result in the release of carboxyfluorocene and an increase in fluorescence. And that's what we see here. So we've got different lipid mixes. So you've got POPC, which is our standard, really easy to use lab model lipid, which is nothing really like a real cell membrane. And then we've got various mixes of different lipids, so POPE and POPG, and POPE and POPC. And these model different types of either yeast or bacterial cell membranes, broadly speaking. And what you can see here is that in each case, as you add more ethanol, you get an increase in fluorescence. And what's striking here, and really useful for our purposes, is you can see that the different lipid mixes have different levels of lysis at the same ethanol concentration. I'm not really going to focus now on which ones are more or less susceptible, but the key point here is that by changing the lipid composition of the membrane, you can change the way that ethanol interacts with this. So what happens if you model that in molecular dynamics? Well, what we can see is the diffusion and the permeability actually align with what we see with our in vitro experiments. So we're looking at a combination of things happening here, one of which is pore formation as ethanol embeds into the membrane, and the other is permeability, which might either be through pores or just through straight diffusion through the membrane. And both those things are important in ethanol tolerance. But what about the membrane itself? What is ethanol doing to the physical membrane? It's really quite challenging to study that in a living cell. So we did a combination of experiments which gave us some information about what's going on. And the first one we did this time was actually the molecular dynamic simulation. So if you simulate a flat bilayer and then you add ethanol to it, what it does is it increases the curvature and the bending and the splaying of the membrane. So in effect, it makes the membrane more spread out and it makes it, for want of a better term, softer. But can you prove that that is happening in a more physical biological system? So we turn to another approach, which is at the bottom, which is high-speed atomic force microscopy. And in simple terms, what you do here is you take a piece of mica, which is very, very flat, and you assemble your phospholipid bilayer on it by adding liposomes, and they fuse. So here is 0 hours, no ethanol added. And what AFM does is it essentially has a tip that dances up and down on the top of your surface, and it tells you how high or low that surface is. And in these images, black and tones of brown are low, so dips, and white is high, so peaks or particles on top of the surface. So in this case, we've got our membrane that we've made and a couple of rogue liposomes that haven't fused, but that's nice to give us some context. Then if we add ethanol over time, what you can see is you get pore formation. So you're getting dips forming in the membrane, actually holes forming through it. And if you measure the height of those, they're the height of the membrane, so the pores have gone all the way through. And the pores get bigger over time. And if you go to 2 hours, what you see is this complete mess. And the challenge here is that you do see pore formation, but the tip itself actually kind of drags across the membrane as it taps. So you see these horizontal streaks as the tip deforms and disrupts the membrane, which is because the membranes become softer. If you don't add ethanol and scan it for 2 hours, it looks exactly like it looks at 0 hours. So the ethanol is making the membrane softer and more susceptible to perturbation. That's great. It's model systems. It's in silico. How do we use that to actually make better strains that you can use industrially? We've got a whole library of strains. We screened maybe 200 in this project for different ethanol tolerances, and we selected 4. They're all code named because they're really industrial strains, so I apologize for that. But what you can see here is a plate-based assay. So you make agar plates with different amounts of ethanol in. You do spots of diluted yeast. So you might have maybe 1,000 or 10,000, 1,000, 100 yeast cells that you start with. And then as you spot out this dilution series, what you can see is in the case of, for example, MY26, although at 0 hours, you've clearly got the same number of cells, they're all gone by 14% ethanol. So 0% ethanol, same number of cells, 14%, nothing gross. So it's tolerant up to about 10%. Whereas these strains at the top, AJ4 and MY14, are much more resistant and can grow up to 18%. So we did that simple screen to give us some tools to investigate the membrane properties. So we selected these 4 strains as being characteristic of either tolerant strains at the top or less or intermediate tolerant strains towards the bottom. And then there's a picture up there just to remind you in the top left of what we're looking at. And we did another assay, which is based on a molecule called laudan. And laudan is a molecular probe. It integrates into the cell membrane, and it's got essentially like an acyl chain, so it sticks in the cell membrane, and a fluorescent head group. And laudan is often said to report on the fluidity of your membrane. What it actually measures technically is the polarity of the head group environment. So if you go back up here a bit, as your membrane displays, you end up with more water accessibility around the head groups, so it becomes more polar. So the laudan becomes differently fluorescent. And you measure the laudan at 2 different wavelengths, do a bit of maths, and you can measure something called the generalized polarization. And confusingly, a decrease in generalized polarization means an increase in fluidity. So if we start with MY26, so this is the strain that isn't really very ethanol tolerant at all, at either 0, 6 or 10% ethanol, you see very little change over time in the fluidity of these cell membranes. Cell membranes do that. Cell membranes have a homeostatic mechanism where they'll change their composition to retain the same fluidity. However, what we see with AJ4 is that at 10% ethanol, it's actually much more fluid. And that's not what we expected to see. We know that ethanol fluidizes membranes, but we expected cells that are more resistant to be able to better modulate that fluidity and retain that fluidity more. But it turns out that cells actually seem to want, in terms of ethanol resistance, to be able to cope with more fluid membranes. How are the membranes more fluid? We've got a whole load of lipidomics data that was done by mass spectrometry that I'm not going to show you because there's slides and slides and slides of graphs. And actually, the thin layer chromatography we did initially tells you basically the same story. Thin layer chromatography is a technique where you take a hydrophilic surface, like silica, for example, and you spot onto that your lipids that you've extracted in organic solvent. So, for example, here we've got some standards, and we've got 0, 6, and 10% ethanol for AJ4, and 0, 6, and 10% ethanol for MY26. And then you can look for changes in the cell membrane at different ethanol concentrations. And each different spot represents a different lipid, and they're characterized different hydrophobicities because you migrate this in an organic solvent. And we use a very simple copper sulfate stain, which stains everything in the cell membrane, basically. And that showed us that we've got a real difference with this one particular lipid. where it's clearly there in AJ4 and clearly not there in MY26. I can't tell you what that lipid is because we haven't published it yet, but it's a very interesting potential correlation between the lipid membrane and the fluidity and the tolerance. This one I can talk about. So what we see here is exactly the same experiment, but this time we're using a specific stain called ninhydrin, and ninhydrin interacts with amino groups, so it only stains a couple of different lipids, phosphatidylethylamine and phosphatidylethylamine, and phosphatidylethylamine migrates here, and what we see is a decrease with increasing ethanol of the amount of PE in the membrane. So we've now got strains that we know are differentially tolerant, but both are seeing the same change in PE. So maybe there's something common here that tells us about the importance of PE within the membrane in terms of ethanol tolerance. So we went back to our molecular dynamic simulations, and to hopefully simplify a very complicated looking graph, what we've got is a situation where we've got a balance of either POPE and POPC in the membrane, and we go from more PE over here to more PC over here. So what you can see is more PE decreases the area per lipid, so you're packing your lipids more tightly together, and therefore, and kind of logically, it also decreases the permeability of your membrane. The other effects we saw from PE by doing our other models is that more PE decreases permeability, which I've just shown you. It also decreases the absorption of ethanol, the amount of ethanol that sticks to the surface, and it also decreases the fluidity of the membranes and their area. So if you'd have asked me at the start of the project, I'd have said, oh, that's good, more PE is what we need. We need decreased permeability, we need decreased absorption, we need decreased fluidity. It turns out that's not actually true. What we see is a complete opposite. So we see that PE decrease with increasing ethanol. So cells seem to be trying to become more fluid to become tolerant. And if we go back up briefly to this Goddard data, that's exactly what we see here. The more tolerant strain has a more fluid membrane. So cells are actually trying to maintain an environment in which they can have more fluid membranes. And that's probably doing a couple of things. The first of which is it's allowing more ethanol to diffuse out of the cell. So the ethanol that's made inside the cell can diffuse out more readily to reduce the intracellular and membrane toxicity. And it's also probably more accommodating of these pores. Because you've got the ability for your lipids to move around more, they can close up the pores more rapidly. So I'm just about to time, I think. So to give you some brief conclusions, as I conclude with most of my talks about lipid membranes, they're really complicated. It's really difficult to understand fully what's going on. But in this case, PE seems to be important due to a range of biophysical properties. So it's not just one thing it's doing, it's modulating the membrane in a very holistic way to allow it to function. And I haven't got the lipidomics data, but it completely backs this up where we see specific PE molecules changing with ethanol concentration. And I think what's really nice about this project and a way of looking at improving cells and biotechnology process is to use that combination of in vitro assays and kind of in cell assays as well, and coupled to in silico experiments, because there's things you can't understand really by doing just an in vitro assay. You need that in silico thing to look at the molecular level of what's happening. And what we're doing now is moving on to engineer strains that make different amounts of PE by messing with their lipid biosynthetic pathways to try and rationally make more tolerant strains. Huge number of people to thank. All the ones on the right and the membrane are all the members of the membrane consortium, and the ones in green are the ones whose data I've shown. And then there's a couple of past members of my lab at the top there, Joyce, who was the project manager, and Sarah, who did all the work I've shown today. My current lab are on there as well. And then the collaborators at the bottom, Simon and Lars, were very much involved in doing the AFM with us. And I will stop there. Thank you very much.
Alan, thank you very much for your presentation. I want to remind people that they can put your questions in the Q&A section, which we have already one. It says, thanks for the interesting talk. Regarding the different lipidomics between industrial strains, I'm wondering if you or partner in the project have planned to see genomics or transcriptomics to verify how lipid synthesis metabolism is different.
Yeah, so we've done transcriptomics, and one of the things we found is, without saying anything we haven't published, is that there's a key transcription factor, we think, which is upregulated in ethanol stress and is actually upregulated in AJ4, which was the more tolerant strain compared to MY26. And that transcription factor acts on a range of different genes. And some of those genes are involved in the control of membrane properties, and specifically the saturation and unsaturation. The data I didn't show you today is that you can also mess with the fluidity of the membranes by changing the degree of saturation of the lipids. So there's some control over that, and also potentially some control over the ergosterol synthesis pathway. So ergosterol is the yeast version of cholesterol. So ergosterol acts like a fluidity buffer within the membrane. So yeah, there's some really interesting transcriptomics data as well. But some of the lipid biosynthetic pathways aren't kind of fully defined yet.
So you already have some maybe candidate genes to work on.
Yeah, we've got some candidate genes, but actually, because the membranes have this homeostatic effect, it's really hard to actually modulate them in cells. So if I took a gene that makes PE, for example, is in the PE biosynthetic pathway, and I upregulate it massively in the cell, nothing really happens to the membrane because the cells are really good at turning that on and off. So actually, the most success we've had is using heterologous pathways. So you take maybe, as an extreme example, an E. coli biosynthetic enzyme and you put it in yeast, and then the yeast lack that endogenous control over the enzyme activity, and you can force the lipid membrane more in one direction than the other. But you're always fighting against the yeast trying to have this homeostatic effect as well.
Then so this I don't know if you already started the experiments, but this modification in the membrane, like how does impact in like the growth itself on like in plates or in liquid media?
So if you modulate it too far in one direction, the yeast are very sick for a number of reasons. Partly, I think, membrane integrity. Partly because specific membrane proteins need particular lipids to function and particular sterols to function. So if you imbalance that, you not only change the biophysics of the membrane, you also change the specific interactions of these membrane proteins with the lipid. So you can't tune them too far. But what we have got with our lipid omics data sets is, and I didn't show you this, but if you take almost any of our yeast strains and you expose them to high levels of ethanol, they start with very different lipidomes in their membrane. But as you get more and more ethanol, they all become very common in terms of what the lipidome looks like. So we think we know what the ideal lipid composition is. And what we're really interested in is not necessarily trying to make cells make that composition, but to make membranes with those same biophysical properties. So you can change the fluidity in a number of ways, you can change the charge in a number of ways. But if you know where you want to get to, you can then pick almost easier engineering targets to let you get there.
In the context of this project, what was the involvement of the industrial partners? Were they able to like mass testing upscale?
Yeah, so this project was done between about 2018 and about 2021. So we all know there's a global pandemic during that time.
Yeah.
So what we were meant to do was we've made some modified hybrid strains that were meant to be tested by Laumann in scale up. You can only do it at particular times of year because Laumann make wine during the winemaking season. So when you harvested your grapes, you do it then. And that happens to be exactly when everybody was in lockdown. So we didn't do it then, but we have got ongoing collaborations with them now to test some of our strains, and we're getting them into their kind of pilot scale fermentation to see if they are any better.
So it's ongoing. That's fantastic.
Yeah. So although the grant's finished, especially our Spanish partner at IATA, I have got a long-standing collaboration with Laumann before and after this project. So they're still working on analyzing those yeast.
And this is just like a last question, like as a young scientist, like kind of what was kind of maybe kind of a challenge to assemble a consortium, like just kind of to all the young scientists in the audience to know.
Yeah, so we are very lucky at us, and I've got a very good friend who works here who's our EU and large grants manager, and he is brilliant because he will cold call and cold email anybody in the world and say, we're looking for some expertise in this area, do you want to be involved? And my findings and my advice is very few people ever say no to that. If you say to people, do you want to be involved in this really exciting project in a big consortium where you're going to contribute what you know how to do, and we're going to come out with this amazing thing at the end, very few people turn around and say no. You know, they might say, I'm quite busy, I can only contribute this amount. But what we did really was I'd come up with the project and I'd identified the skills we got at Aston. So we're very good at lipidomics, we're very good at model membranes, but we've got very little experience in molecular dynamics, for example. And I think as an example of how you can use your networks, I did a postdoc at Oxford, and the lab I was in was opposite the molecular dynamics lab that did membranes at Oxford. So I knew all of them. But the challenge with an EU grant is you can't have all the partners in one country. You can't have everybody in the UK and go to the EU and say, we'd like an EU-wide project, but only in the UK, please. So you had to find somebody else in the world. So basically, I spoke to my mate at Oxford and said, who's the best person in Europe to do this? And he said, it's Siewert Jan, you want to go and speak to him? So I emailed him and said, this is what we want to do. I've got your name from so-and-so. Do you want to be on board? And he said, yes. So I think it's all about making sure you maximize who you know, but also don't be afraid to reach out to people. Scientists love doing science, right? And we appreciate that not everybody knows how to do everything. So make sure you've got your own expertise in your niche, but if you need something from somebody else, reach out to them. So the AFM is another example of that. They weren't formal members of the consortium, but it turned out we needed them. And I happen to know Lars through another network I'm involved in, and he'd mentioned AFM once to me. So I got in touch with him and said, do you know anybody who can do high-speed AFM? And he said, yeah, Simon here does that. And then we got a collaboration with them. So it's just, don't be afraid to approach people. And people generally like doing science with other people.
Okay, thanks for the advice. Yeah, just well noted. Okay, so we are past 8 minutes past 10. So I think I'm going to finish the webinar here. Thank you very much to my panelists, Professor Delvigne and Dr. Goddard. Thank you so much for joining and for presenting your projects. And thank you for the questions that we received. And I'll see you everyone in our next webinar. Thank you so much.
Thanks, everyone.
Thank you. Bye-bye.
Bye. Bye.